Hi, Mark Berger. One other option that you didn't discuss, which I think is something a lot of people are paying attention to, is running trials, whether they're a standard randomized trial or a pragmatic clinical trial, nested within registries or cohorts or networks where data is already being collected on a systematic basis and there's curation going on so that the quality of the data might be richer than it would be in wild type real world data, and that they could build the data collection directly into data capture mechanisms that are already present in the population. And I think behind the idea behind the CoreNet, and there are a number of other networks that have tried a little bit of this. So in England, they have done some pilot projects in their, I'm having a middle-aged moment, what is it? GPRD, yeah. Although they haven't really industrialized it. They showed that it's possible, but they really haven't been able to make it into something that happens on a regular basis. What do you think about that as an alternative to the other alternatives you were talking about?
Well, I think, first of all, the example that I presented of a highly simplified cardiovascular trial was really meant to be illustrative for a variety of approaches in which we capitalize on existing data. And I think what you've described with operating within existing registries is, of course, it's extremely promising. And it for me, that's the category that it fits into. I wasn't meant to exclude it at all from the framework, but if I use this again, I will make sure that it's more explicit. I think it's a great option if the registry exists. If you have to create it, though, then it's challenging because registries themselves they're not inherently dramatically less expensive than randomized clinical trials.
It's Mark McClellan. Elliot, it's great to see the range of trial design alternatives that you described and the potential for substantial cost impact. Notice on your last slide, you didn't go back to restating your 90% cost reduction number with going to something that is more of a truly real world and maybe not traditionally randomized approach. I guess the downside, which we're going to deal with hopefully later today in going to these methods, is that there may be some significant costs in terms of the quality of the data obtained with more heterogeneity, if it's not part of a well-standardized, non-wild type approach, and maybe a loss of power from randomization that's not as well enforced or maybe even fully understood. Have you thought about a way of capturing some of the costs of reduced power, reduced precision? This is something we're hoping the framework and the tools we'll talk about later today can help address.
Yeah, I think that it's first of all it's important to recognize the concerns that you've just raised around real-world evidence. I do think we need to move beyond talking about them and try to actually quantify them so that we understand whether or not they're important considerations in a particular setting. My hope, and it may be a bit of a dream, is that if we're really successful in developing medicines that have very large effect sizes, that some of these concerns will become considerably less relevant. And I think that's the I think what the experience in oncology is teaching us. If a medicine in a single arm trial has a response rate, a durable response rate that's 2 or 3 times higher than what's described in a sort of robust historical data, that is probably real, and it won't disappear when a randomized clinical trial is conducted to actually be confirmed. So I hope as a developer that if we choose the right targets, well validated, we understand the biology, and we bring the best modality to bear on them, something that really can have a dramatic impact on the disease course. It will have effect sizes that really don't, that we're not trying to tease out with massive randomized clinical trials, but that can be easily discerned using, the high quality evidence that's generated in practice settings.
My name is John Birch. I'm with the Mid-America Capital Investment Network in Kansas City. I have two questions. One following up on what Mark asked about registries. You've answered about registries, but in general, more broadly speaking, what are the qualities of real-world data that you would like to see out there that would make your numbers even better?
Which numbers?
Well, you didn't have numbers for it, I have to admit. But the reason to think that the costs of drug development could come down even farther by further use of real-world evidence. I mean, you talked about 85, 90%. I guess there was that number.
Yeah, I think, well, there's no question that that if we, in settings where we can use real-world evidence in place of randomized trial data, that there are dramatic savings that are realized for that particular exercise. I think the unanswered question is how many studies, how many questions will we be able to answer using randomized clinical trial data? It's wonderful to see savings of 85 to 90%, but if you can only apply that rate of savings to 1% of your product portfolio, it's not going to materially alter the costs of running an R&D development engine. And so I think the great question here is, to what extent will we be able to use real-world evidence more broadly? That'll never be applied in every setting. There'll continue to be an important role for randomized clinical trials, but can we use it broadly enough so that it really does begin to bend the cost curve? And that's the kind of answer that's going to come out of workshops like this that help us to understand the challenges of working with real-world evidence and help to develop standards for their application in a regulated setting.
I guess it does go to the question of registries a little bit. Then again, I cite the TASTE trial, which is so impressive, in that it reduced the cost of a clinical trial from roughly $14 million to maybe $500,000. And it seems to me that is something that can be done in more and more diseases, more and more sort of product category. This will be a subject for later conversation, I guess, too, but I just thought I'd mention that. I think it does seem to me that there are categories of real-world data, real-world evidence that could be developed that would apply to be more and more fit for more and more purposes as we go on. But I agree that's the discussion that we're all here about today and tomorrow. Could I just ask you a simpler question? Do you foresee a time when we could say to patients that if you are willing to contribute your data to science, that you will be, and let's just say you even get paid for it, that you will actually be helping to bring the prices of drugs down? You get paid to bring prices down. Can you foresee a day when we can legitimately say that?
I think you've stepped into an issue that makes real world evidence look simple. So,
a question for Elliot. When you talk about moving the research out into the real world, reducing the intensity and burden of data collection, allowing treatments to be delivered by community providers, and the dramatic reductions in cost you would see, my experience is that the regrets come with the null result. Because when you see the null result, that's when you say, I wish we'd measured this, I wish we'd measured that, I wish we'd measured that, because we might learn why it didn't work. But I'm curious, from your perspective, is that really true? Would you say that we will learn less from failure if we don't collect as much detailed data, or would you say failures will still be just as instructive?
I think you're describing a pattern of behavior that was established in the 1980s and 90s when any new drug could be profitably marketed. But today, really, in order to get a foothold in the marketplace, a drug needs to be markedly better than what's currently available. And I think we all have the belief now that if in a well-designed trial a product fails, that it even if we'd been able somehow to tease out an effect through tighter controls on the study, or if we'd measured more data points, that it still wouldn't have been good enough to make a difference for patients. And that, I think is an accurate reflection of today's reality. And so we really have to be prepared. We have to be focused on drugs with large effect sizes and prepared to develop them in a way that allows for failure if they don't have a large effect size. Yes.
Greg Pappas, FDA. I want to thank you, and I was very curious about how you came to your 90%, because it's incredibly close to a figure we came up with where we looked at the cost saving. Actually, we calculated return on investment of doing linked registry data, we call them CRNs, the coordinated registry networks. 23 decisions were supported were made by the FDA using data from this mechanism,
yeah.
And it's about 85% cost savings and years of time savings too, depending on which of the study you're talking about. So I was kind of recalculating in my head. My goodness, it's the same number.
Yeah.
But this is actually an evaluation, a measurement of value of some actual studies that were done. Just curious where
Yeah.
you're at with that.
Well, these are examples that of real-world evidence studies that we've considered, and the assessment has gone so far as to price out a randomized clinical trial, and to also assess the cost of a real-world evidence study. So these are
Projections. These are making projections based on your historic experience with costs.
Well, these are actual experiments, and they would, if they were to move forward, these are the costs that we would be carrying in our budgets.
Thanks.
Elliot, that is always a very thoughtful presentation, and I keep waiting for you to say something I don't agree with, and you did.
Oh, okay.
I got immersed in cancer.
I'm making progress.
The last 2 weeks, and your confidence in these large effect sizes for real clinical outcomes in large populations. We'll discuss over beer or something.
Okay.
But that's the only thing you said that I'm concerned about, because I think most therapies are still going to give modest effect sizes, and particularly for the common chronic diseases that cause 80% of the death and disability that are occurring in the world. We've got to still deal with that. That doesn't nullify anything else that you said.
Yeah.
What I'm interested in, really, for the whole panel, you did a beautiful job of describing what I would call the mentality, which is the right one for how you get a drug developed to the point it gets on the market. And your depiction of changing the types of studies that are done, the design of studies to reduce sample size, et cetera, is exactly at odds with what has to happen after a drug gets on the market, where right now everybody's lost. And it's fascinating to see in cancer now, with these, as more effective therapies are developed, now you've got the same problem in every other disease. Which ones do you use, and how do you combine them? And it goes on. And if you talk to patients, that's the thing, they want cures, but they also want to know what the best treatment is out of alternatives. So how do you stitch? Is it really going to turn out being two different worlds that need to be somehow synthetically put together, or is there a connection between the mad rush to reduce cost by reducing sample size for precision medicine and the need that people have to know which treatments are better for big populations? It'd be interesting to hear from all three of you on that, because I know you're all thinking about it.
I'll let you go first.
I think there's an opportunity for us to stop thinking about a start and stop of a trial. So really moving to the world of continuous trials. So you might have a randomized trial, but it continues, and patients are continued to share their data beyond the trial. And I think that would start to, and as the product moves into the market, you continue to let patients share their data, and that will continue to let us do real-time analysis. I think the technology is starting to be there to be able to do that, and the social behaviors are starting to be there to let us do that. So maybe that would be a way in which to be able to tell what works best for and get to that world of personalized medicine.
Probably need to cut it off here because we're going to get to our break. We're going to get to our break time. So I want to say thanks, everybody. You can probably ask the panelists your questions if you have any. We want to reconvene at 5 after. That's really only about 15 minutes. And thanks to our panelists.